So I want to thank you for the kind introduction and for inviting us here today to talk about Roche sequencing. So I'm representing the Roche sequencing unit that's been transferred to Pleasanton, California, and I'll be talking a little about that in a minute. But I'm the head of healthcare and key opinion leader management for Roche sequencing. And today what I'd like to talk about is the Roche sequencing horizon and a little bit about Nimblegen target enrichment. So my talk will be divided into three parts. First, I wanted to talk about Roche. Roche is a pioneer in next-generation sequencing and underscore some Roche 454 firsts. Then I would like to update you on what's going on with the Roche sequencing unit and many of our collaborations and acquisitions that you might know of over the last year. And then finally, I'd like to conclude about discussing some of the products, the CCAP-EZ, the CCAP-EPI, the just newly released CCAP-RNA, and what we believe is also a very exciting product that will be available next year that we're calling HeatSeq. So as you know, Roche has been quiet over the last few years with the phase-out of 454 Life Sciences, but Roche has always been an innovator. And not only has Roche been an innovator, but they want to bring you the next disruptive, next-generation sequencing technology. And just as evidence of this, I wanted to go through some achievements of 454, and I've actually been involved with Curagen and 454 for about 12, 13 years. And Roche acquired us about 7 years ago. But now we have over 3,500 peer-reviewed publications, and as was the earlier Illumina speaker mentioned, this was the first next-generation sequencer launched onto the market, the GS20. Stefan talked about some of the work that he did with the Mammoth genome. That was predominantly done with 454 sequencing, and 454 sequencing also introduced the concept of ultra-deep sequencing, not only in human cancer, but also in infectious diseases like virology. And you can see how we've gone through different read length advancements, how we've sequenced the first human genome with next-generation sequencing, and also with the release of the GS Junior, which was actually the first benchtop sequencer. Now, I just wanted to briefly touch upon the Junior. The Junior is still very active in the world with the microbial genome sequencing, 1618S, viral deep sequencing, in particular HIV and hepatitis. And I just wanted to point out that this will be supported until the end of 2016. So you'll be able to buy all of the reagents for the GS Junior, and I'll talk about how we plan to bridge that with a smaller PacBio instrument in a minute. So now I'd like to talk about some updates on the Roche sequencing unit. As you know, 454 is located in Branford, Connecticut. I work out of the New York City, Connecticut area. The other companies that Roche has bought, for example, Nimblegen is in Madison, Wisconsin. But the new sequencing unit is going to be located in Pleasanton, California. That's been done for several different reasons. First of all, it puts Roche next to Roche Molecular. So now Roche Molecular can help to develop IVD content for our next generation. It also puts us very close to PacBio, as well as Genia, which are both located in the Bay Area. Now, how does Roche look at the next instrument or the next disruptive technology? And I put this slide on here to just show you our different collaborations and acquisitions and how they complement Roche's version of a complete workflow. So in the next technology that we want to introduce, we know that it needs automated front end. We know that it needs an easy target enrichment technology and single molecule library prep. We understand, and what we want this to be is unbiased single molecule sequencing. We want this to be through a single nanopore. And we also realize that you, the customer, wants downstream specific software. Not only pipeline type of software, but analysis for specific disease indications or for, say, metagenomics. And you can see how these acquisitions and collaborations line up with the complete workflow. And I'm going to talk about these in a little bit more detail. Genia, PacBio, and Stratos provide single molecule library preps. Genia and PacBio are providing single molecule sequencers. Garvan Institute and AbVitro provide upfront workflow and content, and I'll talk about how they contribute to this in a minute. The first collaboration and partnership I want to underscore, and I know Stephen Turner will be doing a keynote talk tomorrow with PacBio, but Roche has developed a very strong collaborative partnership with PacBio to deliver a diagnostic, smaller sequencer. So this system will be based on the Pacific Bio smart cell technology using their waveguides. In this project, we've just had an announcement. This is on track and in development. We hope to bridge the phase out of the GS Junior instrument with PacBio. And if you go to the PacBio website, you can see many of the applications that are being done on the Junior, such as HLA sequencing and HIV are easily done on PacBio. There's also been some very interesting data about how one can further complete genomes with PacBio or sequence very difficult sites of the genome, for example, Fragile X. Genia. Now, Roche acquired Genia in June of 2014, but I'm very excited about this technology because this is a single molecule semiconductor-based technology. It's a CMOS technology where basically one paints a semiconductor with lipid and then can integrate a nanopore. In particular, this uses a hemolysin nanopore, and with the nanopore is tethered to a polymerase. Intriguing about this technology is each of the nucleotides has what's called a nanotag. And the nanotag amplifies the signal as it goes through the nanopore. So we believe this overcomes a lot of the sensitivity specificity problems that you see with a nanopore. This also sequences on the order of milliseconds. It sequences single molecules. Touching on Stratos, why did we develop a collaboration with Stratos? Well, if you look at PacBio sequencing at the surface of a nanopore, or if you look at Genia as sequencing where the cleaved nanotag goes through the nanopore, the Stratos technology actually is a way to derivatize or incorporate derivative bases into DNA by what they call sequencing by expansion. And here, literally, the single molecule DNA passes through a nanopore, and each of the individual bases are amplified by being derivatized by its sequencing by expansion chemistry. So this is true single molecule sequencing. We believe that this will overcome some of the limitations of current nanopore sequencing technologies. And again, it's very fast, accurate, and we believe it'll be low cost as well. The Garvin Institute, this is a technology for applications for Roche to study epigenetic content. Ab vitro is a novel primer extension technology that we've developed a technology acquisition with from George Turch's group. And the benefits of this technology, you'll be able to do primer extension and development of the templates directly either in cell lysates or out of serum without further library prep. And it complements Roche's recent acquisition of another company in Boston called IQM. So this can basically support next generation seeks directly from blood or biologic samples. So at the end of the day, this makes for a complete vision of a workflow. And as you can see, Kappa Biosystems and Ab Vitro are involved in sample prep. Nimblegen, Stratos, and Garvin contribute target enrichment. We have a portfolio with PacBio and Genia to develop sequencing systems. And now, as you can see, we are looking at different analysis tools, software, and diagnostic applications, and look forward to further announcements in the future. Now I would like to move into the third part of my talk and talk a little bit about the CCAP technology. CCAP-EZ is a target enrichment system. This fits very nicely on the Illumina types of system, but CCAP-EZ can be used for whole exome. It can also human, it can also do other species, but it can also develop panels or other areas of interest. The CCAP-EPI target technology has just been released, and this utilizes bisulfate-treated DNA that assays over 5.5 million CPGs, more than 100,000 CPG islands. This differentiates itself from other bisulfate enrichment technologies where this actually captures after the bisulfate treatment. And now we've also launched what we call a CCAP RNA target enrichment system, and this enables you to more efficiently sequence your transcripts. Now, what is CCAP? As many of you know, CCAP is essentially the ability to enrich your DNA. And what you do here is say start with your target DNA. The target DNA is purified. It's cleaved. Here we're putting on Illumina sequencing adapters. You incubate it with your biotinylated sequence capture probe that are specific for, say, your target region or, say, for the human exome. You bind the biotinylated beads with that are captured. You elute the unbound DNA, and now you're ready to go into next generation sequencing. And Roche actually collaborates and sells the Kappa library preparation kit. so that you can put effectively Illumina adapters as well onto the Nimblegen protocol. I won't go into detail on this, I'm just providing the reference, but this is a typical target panel that was developed by Newman et al. It does around 520 cancer exons at 10,000x on an Illumina sequencer. It follows different fusion proteins, and there's a trend now to use circulating nucleic acids in this. But this is a very elegant example. We work, for example, also with Sloan Kettering that has a 250 cancer gene panel with Illumina. Now, I just wanted to talk and go into what we're doing with the CCAP RNA, and this is a way to enrich for your ability to sequence particular transcriptomes of interest. So you make your cDNA of your transcript, it's cleaved by nebulization, it's enriched just like the other sequence capture protocol, EZ, that I mentioned. You amplify your DNA, and now again you can put this readily onto an Illumina sequencer. Now, why does one want to do this? So actually, as you may know, many housekeeping genes, other genes, structural genes, actin tubulin, might be more than 40% of your RNA-seq sequencing. So if you just make a cDNA library and sequence it, why do you want to waste your time sequencing housekeeping genes or, say, for example, actin and tubulin? Well, what you can do with the CCAP RNA then is to make a panel that's specific for just the target transcriptomes that you would want to sequence. So this enables you to do the gene panel profiling, de novo transcript discovery, also go into discovering disease-related transcripts. So it's dramatically increasing your sensitivity and your specificity, say for different regions of the transcriptome that you would like to study. It also is very quantitative and inclusive. So I provided an example here of a way that one could develop, say, a 50 loci gene panel. And this covers the entire loci of, say, 50 genes. So what you're looking at then are the exons of these genes and the non-coding regions. And the question you're asking, are any of these non-coding regions or intronic regions actually undiscovered exons that are spliced in the transcript? And indeed, this paper, Mercer et al., showed that there were approximately 204 unannotated isoforms of these protein coding loci. So 50 loci, about 1 meg of DNA, cDNA on Illumina GA2. And these p53 isoforms then were involved in tetramerization as well as transactivation. So this is a very cost-effective and efficient way to eliminate the transcripts or the RNAs that you don't want to sequence and enrich those that you do. And again, as I pointed out, they have now 204 unannotated isoforms. And these are probably very important isoforms of p53 that need to be functionally characterized. So now, what is available to the customer? So as I mentioned in the earlier paper here, you can basically make a human design, or you can make small, less than 7 meg, or large 7 to 100 meg panels, or 2 different human loci to look at the intron exon splicing. Now, you can also basically have a developer, and I've worked with many different ag bio companies, for example, to look at the different transcriptomes as well. So this is for other human species. But currently out of the box right now, we have a product. This is a long non-coding enrichment kit, and this actually goes across and does 17 or more million base region of human HD19. So this represents about 32,000 long non-coding RNA variants, or regions that are spliced out in the genome. So the idea here would be that you could test your gene transcripts and look at these regions in the non-coding RNA regions to see if you indeed have novel isoforms or splice variants of your genes. Now, my concluding slide here shows a product that we're developing that's coming in 2015. This product we're calling HeatSeq, and it's for high amplification of targets for sequencing. We believe this is going to be rapid, cost-effective. It's going to have a much greater sensitivity and specificity than amplicon sequencing. The HeatSeq probes are advanced versions of MIPS, advanced versions of these molecular inversion probes, and Nimblegen just presented a MIP panel, a greater than 5,000 MIP panel at ASG in November of 2014. And I just want to quickly go through this protocol. So the HeatSeq probes now have a 5' extension primer, and a 3' ligation primer connected by a linker. So the idea here would be that this probe is highly specific in annealing to your genomic DNA, or for example, your cDNA, because it's going to anneal through 2 specific sites at the 5' and the 3' end. So here it's showing the heat-seek probe binding to your target genomic DNA. You do the primer extension as well as the ligation, and then you can purify this DNA based on your target sequence. And for example, if you use universal adapters, either to, say, Illumina or to PGM, you can put this right on to the sequencers. So I want to conclude here by saying that we have a very nice product that we're going to be introducing in 2015. It's the heat-seek product with the 5' and 3' adapters with a linker, and it also has a universal degenerate identifier so that you can study copy number directly from the initial ligation and extension. So with that said, then, I want to thank you for your attention, and I hope that I've provided some insights now, not only into the new Roche sequencing unit, but the Nimblegen CCAP products, as well as some of the products that we're introducing in 2015. Thank you.
Any questions? Yeah.
So you're talking about this heat-seek product that's going to be introduced. Can you tell us more about the economics of it? Because I think that's where it's going to be transformational. Many of these technologies exist, but are more expensive to do in a high-throughput fashion.
Right. So I really can't come up with how we're going to cost it as price. I would assume that it's going to be competitive with amplicon sequencing, because that's what it's essentially made to increase the sensitivity of amplicon sequencing. So if you look at the ASG poster that they did with this cancer gene panel, they compared Illumina MiSeq sequencing done with amplicon sequencing or done with the MIP, and here they clearly achieve sensitivity over background in the range of around 0.1 to 0.5%. And they've also shown that they've been able to use this to detect fusion proteins as well in cDNA, like EML4-ALK.
And can you comment on the extent of multiplexing that would be possible there?
Yeah, so that's a great question. This showed that we had 2 cancer panels that we showed at ASG. One did about 60 genes and one did several hundred, and I only chose to present the 5,000 MIP panel. But there's been proof of concept where one can do a whole human exome from this technology. It hasn't been done by us. It's been done in a peer-reviewed third-party publication.
Sure. Just one question. So are you doing anything to reduce the hype times for the CCAP?
Yeah, okay. That's great. I'm glad you asked that question, because one of the things I wanted to point out today is that we have a protocol for a 16-hour hybridization time with the CCAP. And that protocol's been out there now for about 2 to 4, I think it's about a month. It just came out. Okay. And the idea would be to shorten all these hide times and the heat seek such that it's complementary to a sequencer like PacBio that can run in about 90 minutes or so. And the Genia, we expect to have these similar fast run times. So at the end of the day, we have basically a workflow that you can compete with PCR.
Great. No more questions. Thanks, Bill.
Yeah, thank you.
Yeah, just a moment.